Continuing on from the last lecture, we will now discuss phase 2 metabolic reactions. As we said previously, these involve addition of endogenous groups to chemicals or its metabolites. Endogenous groups include carbohydrate derivatives, sulfate, glutathione, and amino acids. They bind to functional group chemicals or its metabolites. This renders the whole molecule more polar and less water soluble. These are mainly cytosolic reactions. We will now discuss each one of them individually. So glucuronide formation is a major conjugation reaction and involves the transfer of glucuronic acid. The activated form is uridine diphosphate glucuronic acid, or UDPGA for short. UDPGA is formed from glucose 1-phosphate in the cytosol in a two-step reaction illustrated at the bottom of the slide. Firstly, it involves the addition of UDP, and secondly, it involves oxidation to form UDPGA. The final part of glucuronidation involves the conjugation of UDPGA to the target molecule. The conjugation reaction is catalyzed by UDP glucuronyl transferase, or UDPGT. These are microsomal enzymes, meaning that they're in proximity to the smooth endoplasmic reticulum and consist of two gene families, UGT1 and UGT2. This reaction increases the aqueous solubility of a xenobiotic. Many products become recognized by the biliary and renal organic anion transporters, which we encountered in the first lecture in the series. Glucuronides are also substrates for intestinal beta-glucuronidase enzymes produced by bacteria, leading to something called enteropathic recycling, which we will discuss in the next lecture. Shown in the diagram is an example of such a reaction with paracetamol, with UDPGA being added to the hydroxyl group at the bottom. The second reaction we will discuss is sulfate conjugation. Similar to glucuronidation, it involves two steps. The activated form transferred is called PAPS, or phosphoadenosyl phosphosulfate. PAPS is formed from sulfate and ATP in the cytosol, involving addition of ATP and phosphorylation as shown in the diagram on the right. Conjugation reaction is catalyzed by an enzyme called sulfotransferase, a cytosolic enzyme, and typical substrates include phenols, alcohols, and N-hydroxy compounds, and paracetamol is illustrated as an example on the bottom right of the slide, where the PAPS molecule is added to the OH group at the bottom, as seen in the diagram. The third reaction we will discuss is acetylation. This is conducted by a cytosolic enzyme called N-acetyltransferase, or NAT for short, using acetyl coenzyme A. There are two major forms of this enzyme, NAT1, which is found in most tissues, and NAT2, which is mainly found in the liver and is polymorphic, meaning that there is variation in the general population for its genetic sequence. Slow NAT2 acetylators are predisposed to drug toxicities, such as peripheral neuropathy from the drug isoniazid, for example, which is used to treat tuberculosis. Typical substrates include aromatic amines, sulfonamides, hydrazines, and hydrozides. These enzymes detoxify aromatic amines by conversion to amides that are less likely to be DNA-reactive metabolites. This reaction does not enhance water solubility greatly and may even reduce water solubility in some cases. Glutathione, or GSH, illustrated in the diagram on the top right, is a tripeptide, meaning that there are three amino acids consisting of glutamic acid, cysteine, and glycine. It's found in most cells and is abundant in the liver. The sulfohydryl, or SH group on cysteine, is nucleophilic and reacts with electrophiles, e.g. reactive metabolites. Glutathione conjugation is an enzyme catalyzed reaction involving glutathione S-transferase and is a direct chemical reaction. It is mostly a cytosolic enzyme with multiple forms and has different substrate specificities involving the transfer of GSH to electrophiles. Shown in the example on the right, you can see glutathione transferase transferring a glutathione molecule to a substrate, in this case an epoxide. Typical substrates for glutathione conjugation include epoxides, both aromatic and aliphatic, best known are as being present in glues and solvents, and also some halogenated unsaturated compounds and alkyl halides. Some examples are given in the diagrams on the right, including dichloronitrobenzene, bromobenzene, and our worked example paracetamol. This process can involve conjugation reactions that can be divided into displacement and addition reactions. Displacement is where glutathione actually displaces an electron withdrawing group, such as shown in the diagram on the top right and in the middle. And addition reaction is where glutathione is added to an activated double bond or strained ring, as is the case in the addition of glutathione to paracetamol. Mercapturic acid formation is a major result of glutathione conjugation, and the reactions to form it involve the sequential removal of amino acid residues from glutathione, as is shown in the diagram on the right. Firstly, removal of glutamic acid involves glutamyl transferase. Removal of glycine involves cysteine glycinase, and finally it involves an acetylation reaction using N-acetyl transferase of cysteine in the last reaction. Main sites of formation of mercapturic acid are the liver and the kidney, and once it's formed, it's excreted in the urine or bile. Our next pathway is methylation, which is a minor pathway in biotransformation and may even lead in some cases to greater toxicity. It often decreases the water solubility and then masks some functional groups that may be conjugated. Methyl groups are transferred by N, S, and O methyltransferases. An example of such a reaction is depicted in the diagram on the right, where mercaptopurine is methylated on a sulfur group. S-methylation in this particular example is mediated by an enzyme called TPMT, or thiopurine methyltransferase, and it occurs in the microsomes. Amino acid conjugation of xenobiotics containing a carboxylic acid group involves addition of an amino group of an amino acid. Illustrated in the diagram on the right is conjugation with glycine, glutamine, or taurine. Typical substrates therefore include aliphatic, aromatic, and aryl acetic acids. Conjugates are eliminated mostly by urine. A good example of a substrate is benzoic acid or benzoate. As illustrated in the diagram, it involves an activation step by conjugation with CoA by acyl-CoA synthase, followed by a reaction with an amino group catalyzed by the enzyme acetyl transferase. If we return briefly to our worked example for the PET toxicant, we now have encountered multiple pathways that affect paracetamol, including glucuronidation, sulfation, and glutathione conjugation. In the current slide, we can see that paracetamol is affected by both phase 1 and phase 2 metabolism. These, as we will see, can often have different consequences. Again, returning to our slide on the toxicokinetics of paracetamol, we can see on the graph on the right that there is an increase over time in the concentration of paracetamol's metabolites, some more so than others. It is important to realize that metabolism is not a uniform process and can vary quite drastically between individuals and for different chemicals, even though they might be similar in nature. We will now discuss some of these factors that can influence metabolism in a bit more detail. The first of these factors is the lipophilicity of a compound. I would like to illustrate this a bit better with an example of two compounds that are very similar and yet can be metabolized very differently. Thiopental and pentobarbital are very similar. There is only one atom difference between them, as is illustrated in the structures on the right. They even have a similar pKa, i.e. there is a similar proportion ionized in each case. However, there is a difference in lipophilicity, in that thiopental is more lipid soluble. This has a physiological consequence, as is indicated by the graph on the bottom right. This graph shows the concentrations of both compounds in 3 different tissues over time: the blood, the brain, and body fat. As can be seen, thiopental enters the brain rapidly, giving anesthesia, and is short-acting as it gets redistributed to fat. On the other hand, pentobarbital enters the brain more slowly, giving a hypnotic effect, and is retained more, meaning it is longer acting. A second factor we can consider is the structure and often the chirality of a compound. And to illustrate this, we will look at a classic example in thalidomide. Thalidomide was a drug used during the late 1950s for morning sickness. It was manufactured as a racemic mixture, meaning it had both R and S forms, enantiomers of the compound. This caused birth defects in the children of women who used it for morning sickness. The S-thalidomide is the form that was responsible for the birth defects. Another example of this involving structure, in that minor structural differences may have a substantial effect on toxicity, is that of PCBs, or polychlorinated biphenyls. As is shown in the diagrams, we have 2 compounds with the same chemical formula but very different structures. One of these is immunotoxic, the other is not immunotoxic, even though they both have the same chemicals contained within their compound. In addition to chemical factors, there can be biological factors, in that you can have interspecies difference in toxicity. As we can see by the tables on the right, the same compound can have different toxicity in different species. The 2 tables show the LD50 values for 2 different compounds: TCDD, which is a dioxin, and previnium chloride, which is a rodenticide. Remember, the lower the LD50 value, the more toxic it is to that species, as it means it only takes a small dose to kill 50% of the population. Interspecies differences in toxicity are due to various factors. They can be due to things like distribution, as there may be differences in the amount of fat, or serum proteins, or types of serum proteins. It could be due to metabolism, due to differences in the metabolic pathways, or differences in the microbiome, which can help in transformation in the gut, for example. It could be due to absorption, in part due to differences in gastric pH, and this would be particularly relevant to weak acids. There can also be sex differences within a species. A good example of this is with glutathione conjugation in mice. As we have seen already, glutathione is a tripeptide with the 3 amino acids, glutamic acid, cysteine, and glycine, found in most cells and abundant in the liver. One enzyme involved in synthesis is glutamate cysteine ligase, or GCL. GCL is higher in female mice than it is in male mice. It has been found experimentally that female mice replenish glutathione faster than male mice after paracetamol poisoning. Mouse glutathione levels are highest in the morning, as they've spent most of the night eating, being nocturnal animals. For example, a 300 milligram dose in the morning is much less toxic than a 300 mg per kg dose in the evening. During the metabolic process, there can be enzyme inhibition, particularly with enzymes of broad substrate specificity like the CYP P450 family. If an important enzyme in the metabolism of a toxicant is inhibited, it can mean an increase in the concentration of that toxicant. As is shown in the graph on the right, this can mean an increase in concentration into the toxic range. This can involve different types of inhibition. Competitive inhibition happens when 2 different compounds are binding at the same active site of the enzyme reversibly. Non-competitive inhibition can occur when there is binding at 2 different sites. This can be an important concept in preventing paracetamol and ethanol hepatotoxicity. For example, diallyl sulfide inhibits cytochrome P452E1 activity, which means it inhibits the generation of NAPQI, which, as we saw in the last lecture, gets generated during paracetamol metabolism and is a toxic metabolite. This slide illustrates another example of enzyme inhibition and a new concept called synergistic toxicity. The photo shows the ink cap mushroom, which itself is edible and not toxic, but it contains a chemical called coprin that causes a sensitivity to alcohol. The reason why this happens is that it inhibits acetaldehyde dehydrogenase, as is shown in the equation at the bottom, involved in the metabolism of alcohol. This means that there is an accumulation of acetaldehyde, which is the toxic metabolite from the first reaction involving alcohol dehydrogenase. We also see an increase in enzyme activity caused by a foreign compound. This can often require repeated or prolonged exposure to something called an inducer. Generally, it involves increased gene transcription of the enzyme. As is shown by the graph on the right, enzyme induction may increase or decrease toxicity depending on whether the metabolite or the parent molecule is more toxic. A number of different chemicals are known to be inducers. For example, phenobarbital type inducers cause a wide range of metabolic enzymes to be increased, including the cytochrome P450 enzymes. Dioxin type inducers cause an increase in protein levels of specific cytochrome P450s, and also an enzyme called aryl hydrocarbon hydroxylase. We will now look at a specific example of where we can see induction. Benzoate pyrene is present in all smoke, resulting from the combustion of organic material, including cigarette smoke, barbecued food. It's also present in coal tar and in car exhaust fumes. It is what is known as a pro-carcinogen and is involved in a wide variety of metabolic transformations. There are 3 reactions involved in activation, as is highlighted in red in the pathway on the right, with the ultimate carcinogen being something called dihydrodyl epoxide. Benzoate pyrene is able to bind to something called the aryl hydrocarbon receptor, or AHR. As is shown in the diagram on the left, the AHR is able to translocate to the nucleus, where it binds to DNA in specific areas of DNA called xenobiotic response elements. These are sequences in DNA that are located upstream of certain key genes that code for enzymes. There, they induce the transcription of those genes. An example of this would be cytochrome P450-1A1. And as we saw on the previous slide, CYP1A1 is involved in the metabolism and the generation of the carcinogen dihydrodyl epoxide. So, at least from this, we can conclude that benzoate pyrene induces its own activation. It seems that the loss of the AHR gene protects people from carcinogenesis. Another good example of enzyme induction is the synergy of alcohol and paracetamol. Repeated alcohol in some consumption induces the expression of cytochrome P450-2E1, and as a result of this, increased CYP2E1 increases the metabolism of its substrates, including both ethanol and paracetamol. So alcohol increases the susceptibility to paracetamol-induced hepatotoxicity due to the increased formation of NAPQ1, which is shown in the diagram on the right. As we can see, CYP2E1 is responsible for the generation of NAPQ1 further down in the pathway. Finally, there are some biological factors that can affect individual toxicant metabolism, including diet, disease state, and age. Reduced diet may affect metabolism. It may also decrease toxicity in some cases, for example with aflatoxin, where there is reduced bioactivation. Alternatively, it may increase toxicity, as with paracetamol, as there is reduced glutathione levels after fasting. Sometimes deficiencies in vitamins and trace elements can also limit detoxification of some chemicals. Examples of this include vitamin E or selenium deficiencies. There are many examples whereby toxicants can enhance the pathology of certain diseases. For example, some chemicals can increase the hepatotoxicity of tumor necrosis factor alpha, which is a key cytokine increase during some inflammatory diseases. Age is also a major factor in metabolism. For example, infants have much lower metabolism than adults, they have lower renal clearance, they have altered distribution with a lower percentage drug found in body fat. The elderly, on the other hand, have altered distribution with higher percentage drug in body fat. Another thing to consider is the disease status of an individual, because this can affect their toxicant metabolism. Diseases of the liver, for example, can result in reduced metabolic activity and clearance, reduced protein synthesis, including reduced plasma protein synthesis. This can lead to higher free concentrations of a toxicant in the plasma. Renal disease can result in reduced filtration and reabsorption and higher free plasma levels in the individual as a result. Cardiovascular disease finally can result in reduced cardiac output, reduced metabolism and reduced renal clearance.